Hi, today we're going to take a look at two trait crosses. We're going to also take a look at dihybrid crosses. So two trait crosses or dihybrid crosses are looking at two genes and the allele combinations of two genes. So in this instance, we are going to take a look at the gene for seed color. We're going to take a look at the gene for seed shape, and I've already got spelled out here. The dominant allele for seed color is yellow, represented by a capital Y. The recessive allele is green, represented by a lowercase y. For seed shape, round is the dominant allele and is represented by a capital R, and wrinkled is the recessive allele, represented by a lowercase r. So we're going to take a look at how we construct a Punnett square when we're looking at two trait crosses. So we're going to take a look at a homozygous cross first. So when you cross a pea plant that is homozygous for round and yellow seeds with a pea plant that is homozygous for wrinkled and green seeds. So first off, we want to establish the genotypes of the parents. So in the example here, we see that we have a pea plant that is homozygous, so that means that both of our alleles are going to be the same for round and yellow seeds. So that means that I'm going to have a genotype for my first parent of capital R, because it's homozygous for round, and then it's also going to be homozygous for yellow. So we're going to go capital Y for that. And I'm going to cross that with my second genotype for a plant that is, again, homozygous. So we're going to have the same alleles for wrinkled and then green seeds. So in this case, I'm going to have a lowercase r, followed by a lowercase y, and a lowercase y. Perfect. I'm going to scroll this up. Oops. I think that should be good. So in order to figure out your gamete combinations, or what falls along the boxes here, 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 and here, there's a specific trick that I like to use in order to figure out those gamete combinations. And the trick is this. So what you'll do is you'll draw a line between the alleles for the two different traits, and then you'll write 1, 2, 1, 2. And in doing that, I'm going to show you a quick trick. Once you've done that, you're going to write the numbers 11, 12, 21, and 22 alongside your page here. Now for 11, you're going to go to the 1 on this side, and then you're going to go across the line and go to the 1 on this side. So we're going to have a capital R, capital Y. For 12, we're again going to start with the 1 on this side and go all the way over to the 2 on this side. So we're going to have an R, capital Y. And then for 21, we're going to go for the 2 here to the 1 on this side. So again, capital R, capital Y. And then last but certainly not least, we go 2 to 2, and we're going to do a capital R, capital Y. I know it's white, it's not perfect, but we got our different combinations. And then we're going to put 11 here, so R, Y, 12 here, R, Y, 21 here, R, Y, and 22 here, R, Y. Now, because this is a homozygous, all of these gamete combinations are going to be the same, but if they were in fact different, this 11, 12, 21, and 22 is a good technique to use in order to figure out your gamete combinations. We can do the same thing over here with this one. Again, we can draw a line in between the alleles for the two different traits, do 1, 2, 1, 2, but we can pretty much figure out what the allele combinations are going to be. So they'll be R, Y, there'll be R, Y, there'll be R, Y, and then down at the bottom we'll have a lowercase r, lowercase y. And then you'll just drag and drop your gametes into the proper Punnett square boxes. So again, we'll have capital R, lowercase r, capital Y, lowercase y. Capital letters always come before lowercase, and I like to go in alphabetical order, so the R's will always come before the Y's. So if you fill in all of the boxes within the Punnett square, you will see that 100% of them are going to be heterozygous for their genotype. So capital R, lowercase r, capital Y, lowercase y, and 100% of them are going to express the dominant trait. So they're all going to be round and yellow. Okay, perfect. Next, we're going to take a look at a cross between two heterozygous pea plants. So our genotype, when you're crossing two pea plants that are heterozygous for round and yellow seeds, our genotypes are going to be as follows. So we're going to have a capital R, lowercase r, capital Y, lowercase y, and then a capital R, lowercase r, capital Y, lowercase y. Again, we're going to use that technique that I showed you on the first page, the 11, 12, 21, and 22. So you draw a line between the alleles of the two genes. You write 1, 2, 1, 2. Then off on the side, you write 11, 12, 21, and 22. And then again, 1 to 1, we're crossing that center line. We have capital R, capital Y, 12, 1 to 2, a capital R, a lowercase y this time, 21, lowercase r, capital Y, so a lowercase r, a capital Y, and then 22 is going to be a lowercase r and a lowercase y. So then we put our 11 here, capital R, capital Y, our capital R, lowercase y here. We do our lowercase r, capital Y here, and then our lowercase r, lowercase y here. Now we could do the same thing for this one, the 11, 12, 21, and 22, but we're just going to end up with the same gamete combinations as we did over here, so we can just fill that in. So capital R, capital Y, capital R, lowercase y, lowercase r, capital Y, and then lowercase r, lowercase y. And then you would fill in your boxes by, again, dragging and dropping. So I'm just going to do a brief sample right now of what those are going to look like. So we're going to have, capital R, capital Y's, capital R, lowercase y, lowercase y. We're going to have lowercase r, capital Y, lowercase r, lowercase y, lowercase y. So we can see that we're getting a bunch of different genotypic combinations as we fill in the boxes. Now, every time you do a heterozygous cross, when you're doing a dihybrid cross or a two-trait cross, you are always going to get a phenotypic ratio of 9 to 3 to 3 to 1. Always. 9 of them are always going to have the dominant trait for both alleles. So 9 of them are going to be round and yellow. 1 of them is going to be completely recessive. So this is going to be wrinkled and green. And then what I like to do is I like to kind of fill in. So 3 of them are going to be also green, but they are going to be round and green. And then 3 of them are going to be yellow, but they are going to be wrinkled and yellow. So anytime you have a heterozygous heterozygous cross and you're working with two traits, you are always going to have a phenotypic ratio of 9:3 to 3:1. I will never ask you about the genotypic ratio for this specific cross because, as you can see from these 4 boxes, the genotypes can get a little crazy, but this phenotypic ratio is consistent across the board, so you must know that. I hope you found this helpful. Let me know in class if you have any further questions.